Thank you for viewing this webinar on structural variation detection with NextGENE software. We'll discuss the structural variation detection algorithm, settings, reporting, and we'll review some demo data. NextGENE's structural variation detection algorithm works by first detecting regions where mapped reads contain a high level of mismatch at one end. These reads are split at the location where the mismatches begin and are realigned as pseudo-paired reads to track where each end maps. A consensus sequence of regions where pseudo-paired reads map is also generated. If the sample data is from RNA sequencing, select a reference that includes genomic and RNA reference sequences. Such references are available from the SoftGenetics FTP server for the human genome builds 37 and 38, and these include plus mRNA in the reference name. To perform structural variation detection, the Detect Structural Variation setting should be selected in the project wizard. Then, click SV Settings to specify the settings for structural variation detection. Under Detect Pseudo-Pair, you can define the requirements for detecting potential SV breakpoints based on the mismatch region at the read's ends. Thresholds can be set for the mismatch ratio, the percent of the read that is mismatched, and/or for the mismatch bases, the number of bases mismatched at the end of the read. Note that you must also ensure that the overall matching base percentage setting is set low enough to allow for these reads with mismatched ends to be aligned. For example, if you set the mismatch ratio to 0.1 or 10%, then the overall matching base percentage must be set lower than 90%. Regions with reads aligned meeting these thresholds are considered as SV breakpoints. Under breakpoint filters, you can set further requirements for the breakpoint to be reported. Set thresholds for the breakpoint total coverage, linked reads ratio, which is the ratio of the linked reads over the total coverage at the breakpoint, and the linked reads count. Linked reads are defined as the reads spanning the breakpoint. Lastly, you can select to prioritize alignments to genes. If a read could be aligned to multiple locations, alignments to regions annotated as genes are prioritized over non-gene regions. You can additionally select to specify a gene list in a text file. Alignments to the gene regions listed in the text file will be further prioritized. Once the analysis is completed, you can view the structural variation results by going to Reports, Structural Variation Report. In the dialog that opens, select Structural Variation/Fusion Report. Then you can select to generate the report based on either raw linked read counts or scaled linked reads, which applies a normalization factor to the linked read count, and then set a threshold for reporting. You can also click on the Display and Filter tabs to further define the settings for the report. On the Display tab, select the columns to be included in the report. On the Filters tab, you can select the type of variance to be reported, as well as other filtering options, including whether or not to merge overlapping events, to include unique and/or ambiguous breakpoints, and to filter based on the region type, such as reporting variance only in gene or coding regions. Note that you can click Save Settings to save a settings file for these options. Click OK to generate the report. The structural variation report includes a linked viewer pane above the report that displays the two ends of the structural variant. These are the link reads, reads where one end of the original read maps to the region shown on the left, and the other end of the original read maps to the region shown on the right. For each structural variation reported, the start and end positions of the partial reads on both sides of the relevant breakpoints, as well as both breakpoint positions, are listed. Further, both the start and end positions and the breakpoint can be shown using genomic positions and/or relative to RNA, CDS, and protein numbering. Additionally, the distance between breakpoints, the variant type, and annotation at the breakpoint are included. Output options are shown after clicking the Report menu. The report can be saved in text or VCF formats. VCF output options include saving the filtered report, which will include only the structural variants that meet the filter criteria specified in the settings, or saving an unfiltered report. The unfiltered report will include all structural variants detected, with those that fail the filtering criteria flagged as SGFLT in the filter column of the VCF. You can also select to save the consensus sequence in FASTA format. This FASTA output will contain consensus sequences for the reported structural variations. You can double-click any structural variant in the report to visualize the variant in the top viewer pane. The example data shown here is from the SaraSeq Fusion RNA Mix version 2 from SaraCare Life Sciences, which includes numerous known fusions. Let's take a closer look at one of these fusion events. Entry 5 in the report shows a fusion event between the EML4 and ALK genes. We can double-click on this entry in the report to move the visualization to show the two ends of this fusion. The genomic coordinates for the region covered by the reads at each end of the fusion event are reported, as well as the coding base numbers. Genomic and coding base coordinates are provided for each breakpoint as well. Fusion is shown as the variant type. We can see that one end of the fusion event is at CDS12 of the EML4 gene, and the other end is at CDS20 of the ALK gene. There are 22 forward-oriented and 70,730 reverse-oriented link reads for this fusion event. We can click on the Report menu to view the settings for the report, or to save the report in any of the formats described previously. Thank you for viewing this webinar on structural variation detection with NextGENE software. If you have any questions, please feel free to contact us at tech_support@softgenetics.com.